Welcome back to another video of STEMAC, and in this video we're going to explain DNA replication. So in DNA replication there are some major components that takes part in the whole process. So these are the enzymes and protein that completes the DNA replication process. So at first, helicase enzyme. Now we know that DNA is double-stranded and they are the nucleotide base pairs are bound together with hydrogen bonds. So this helicase enzyme, they are going to break those hydrogen bonds between the base pairs at a replication fork. So in the origin of replication, a replication fork will be formed by the helicase enzyme, as the helicase enzyme will break the hydrogen bonds between the nucleotide base pairs within the double-stranded DNA molecule. So as the helicase enzyme is breaking the hydrogen bond, this region at the front of the replication fork will be stressed, right? And so to prevent the supercoiling of the front region, a topoisomerase enzyme will come and bind here. They will always be at the front of the replication fork, so that a supercoiling doesn't occur in this region. And then we have some single-stranded binding proteins, these black proteins. They are single-stranded binding proteins, and what they do is they keep these single-stranded DNAs. Now we have two single-stranded DNA, right? They keep these two single-stranded DNA into their position, so the replication process doesn't get interfered. So after this, we know that DNA strands are anti-parallel to each other. So if this is a 3' to 5' strand and this is a 5' to 3' strand, the complementary DNA strand will be 5' to 3' for this one and 3' to 5' for this one, right? So these two are now template strand, and along with this these two template strands, we will get two complementary new DNA strands. So first of all, a primase enzyme will come and bind here. Okay, so this is a primase enzyme. And the primase enzyme will bind RNA primer. RNA primer are a few nucleotides long, and they will come and bind with the primase enzyme here. So this is our RNA primer. This is 5', starts from 5', okay? So now this RNA primer has a 3' end here, right? A free 3' end. Now DNA polymerase III will come and bind nucleotides. They will start from this 3' end, because a DNA polymerase III can only start their nucleotide binding, the complementary strand synthesis, from a free 3' end. So we will need, that is why we need a RNA primer to start the replication process, because the RNA primer has a 3' end, free 3' end, where the DNA polymerase can come and bind. So the DNA polymerase, that is why the DNA polymerase will always conduct the replication process from 5' to 3' end. So the DNA polymerase will come and bind here, and they will start the replication, and they will continuously keep binding a complementary nucleotide base pair here. So let's say if this is adenine, okay, they will bind a thymine here. If this is guanine, they will bind cytosine here, right? So this is the leading strand because it's going in the 5' to 3' direction continuously. So we will call this strand the leading strand. Now, in the case of this template, which is going in the 5' to 3' direction, the antiparallel strand has to be 3' to 5', right? But as we learned, a DNA polymerase cannot go from 3' to 5' direction. So in this case, what's going to happen is that a primase enzyme will come and bind RNA primer with the primase enzyme. So let's say here is a RNA primer. And as the replication fork will move forward, RNA primer will keep binding to different places, different segments. So when our RNA primer has bound to a region, the DNA polymerase will come and bind from the 3' end of the RNA primer. So this is 3' end, this is going to be the 5' end. So in this case, what's going to happen is that we will get these fragments. Because the replication fork is moving forward and the RNA primer keeps binding, and the DNA polymerase then binds to the 3' end of the RNA primer. So we were getting this fragmented strand, right? And this is called the lagging strand. This is our lagging strand. And these fragments are called Okazaki fragments. So after this, the DNA polymers will bind the complementary nucleotide base pair. Then comes DNA polymerase 1. So DNA polymerase 1 will remove the RNA primer. They will remove the RNA primer nucleotides. So when the RNA primer nucleotides are removed, the DNA polymerase cannot bind this to, we can see some gaps have formed here, right? DNA polymerase 1 is not capable of binding this or filling these gaps by binding these two nucleotides base pair together. So we will need another enzyme which is called the ligase. So ligase enzyme will come and bind here, and they will attach these two nucleotides together and fill up the gap that was formed by DNA polymerase 1. As DNA polymerase 1 removed the RNA primer, ligase will come and bind and attach. They will fill this gap, okay, by attaching these fragments together. So this is how the replication process is finished and conducted with using these major components.